Before moving on to protein metabolism, I'd like to review the structure of a protein. Proteins consist of repeating units called amino acids. An amino acid consists of three groups coming off of a central carbon along with the hydrogen. An amino group, a carboxyl group, and a side chain. 20 different types of side chains exist. Therefore, there are 20 different types of amino acids. Making new proteins from amino acids is called protein synthesis. There are several different sources of the amino acids used in protein synthesis. Body proteins that have been catabolized is one source, as well as amino acids absorbed across the small intestine mucosa. The amino acids absorbed across the small intestine mucosa come from the foods that you consume, from dead mucosal cells, as well as from digestive enzymes that have digested each other. There are three processes involved in protein metabolism. We will look at each process in greater detail. I'm just going to list them now. Oxidative deamination, keto modification, and transamination. During transamination, the amino group on an amino acid is transferred to a keto acid. There's my amino group. I'm transferring it over to the keto acid from the amino acid. So going from the amino acid to the keto acid. When that happens, the amino acid is converted into a keto acid, and the keto acid is converted into an amino acid. The only difference between the amino acid and the keto acid then is the presence of the amino group. If there is an amino group, it is an amino acid. If there is not, it is a keto acid. This allows me to use different types of keto acids to make amino acids by just simply adding an amino group, and the opposite is true. If the keto acid is alpha ketoglutarate, which recall is an intermediate of the citric acid cycle, I can use that to make an amino acid glutamate. Notice the arrows go in both directions. So I can break down my proteins, have the amino acid glutamate, and convert that into a keto acid. During oxidative deamination, the amino group on the amino acid glutamate is removed. When that occurs, the glutamate then becomes alpha ketoglutarate. Remember, alpha ketoglutarate is an intermediate of the citric acid cycle. So glutamate can be used to synthesize ATP once it has been converted into alpha ketoglutarate. Recall that during the citric acid cycle, ATP is synthesized by substrate level phosphorylation, and NAD and FAD are reduced. Those reduced NAD and FAD will go to the electron transport chain, where the electrons will be used to make additional ATP. The amino group that is removed from the glutamate becomes ammonia. Ammonia is highly toxic. The liver will then convert the ammonia into urea. Remember, oxidative deamination occurs in your liver, so the glutamate that we just produced during transamination will go to the liver, and by the process of oxidative deamination, the amino group will be removed in the form of ammonia. The glutamate, remember, then becomes alpha-ketoglutarate. The ammonia, since it is highly toxic, is converted into urea by the hepatocytes, and carbon dioxide is required for this conversion. The urea then will travel to the kidneys, where the kidneys will eliminate the urea from the blood. If the liver is not functioning, the ammonia will not be converted into urea by the hepatocytes, and this will lead to a hepatic coma, which will cause death within a few days. Let's look at the urea cycle more closely. Recall that glutamate, which can be produced during transamination, goes to the liver. At the liver, the amino group of the glutamate is removed during oxidative deamination in the form of ammonia. The liver cells or hepatocytes then convert the ammonia into urea. This is because ammonia is too toxic to be transported in the blood. Urea, however, can be transported in the blood to the kidneys, where it can be eliminated from the body through the urine. So urea is an end product of amino acid catabolism. The last process I'm going to cover in protein metabolism is keto acid modification. During keto acid modification, keto acids are converted into other molecules such as pyruvic acid, acetyl-CoA, and intermediates of the citric acid cycle. If a keto acid is converted into pyruvic acid, that pyruvic acid then can be used to synthesize glucose or glycerol required to make triglycerides. If it is converted into an acetyl-CoA, the acetyl-CoA can be used to make the fatty acids of the triglycerides, ketones, cholesterol, or additional ATP by way of the citric acid cycle and the electron transport chain. If it is converted into an intermediate within the citric acid cycle, such as alpha-ketoglutarate, it can be used to make additional ATP. So we can take proteins, break them down into amino acids, convert the amino acids into keto acids, and then convert those keto acids into other molecules that can be used either to make triglycerides, glucose, or be used to make additional ATP.